How is it possible that something that used to take like 30 days, 14 days, and now is being done in 5 minutes with the help of just updated sensors and AI?
The accuracy of the FLIM over, since like 1930 is 94 to 99% accurate, distinguishing cancer cells from non-cancer cells. And we received the same accuracy rates on our initial study. This would change that management and make it better for everyone, including the hospitals.
Hello, Rachel. Thanks for joining Scouted by GreyB. I'm very excited to have you here. So how's it going today?
Hi, Shabaz. I'm thrilled to be here. Thank you for inviting me.
Before I dive in, I'd like to ask you, and for our audience, can you summarize, what Caelum is doing? You might have to boil it down into like simple terms.
That's okay, Shabaz. I'm a breast surgical oncologist, and in my decade of practice and my 20 years in the field, I came across a pain point that was interesting. My patients, if they had a suspicious finding on a mammogram or an ultrasound of their breast, sometimes the radiologists, the surgical oncologists, we all agreed that that patient needed a tissue biopsy. The lesion or whatever was found, the suspicious finding on imaging, required a needle to be placed into that tissue and tissue to be removed, just sampled to see if it was cancer or a precancer, a high-risk lesion, you name it, or a benign lesion. Actually, in the breast field, 80% of biopsies are benign and 20% are malignant. So most of these findings are not cause for alarm, but the patients always experience alarm, crippling anxiety. I get phone calls at 2:00 in the morning saying, I know I have cancer, that kind of a thing. And even if the patients did end up having cancer, they were less anxious in general about the diagnosis once they knew, okay, now we know what we're doing. We have a plan. So that was a pain point, and it affected me. I really wanted to sort of solve that problem. So, there is an old technology called FLIM, fluorescent light imaging microscopy. It was discovered in the 1930s, I think, at the Carl Zeiss Institute in Germany, and been refined ever since. And what it does is it uses autofluorescence of the tissue. It excites tissue, healthy cells and cancer cells, or unhealthy cells, even not in cancer. They get excited. They hold on to the excitation from a laser or LED and release it at different time points at different rates. And that amount of time from excitation to release is called the lifetime, the L in FLIM. That is an indirect measure of metabolism. And if you can understand, anyone can understand, cancer are cells that rapidly divide out of control. Okay. So their lifetime is very short. The time to excitation is very, very short. Whereas a normal cell will metabolize and then undergo what's called apoptosis, which is normal cell death. And that's how the body works. So, and when you have cancer, it's a rogue couple of cells that don't get that do not undergo apoptosis, and they just kind of go wild, and then they spread, et cetera, et cetera. So that time to wait ranged, in New York City from a couple of days to a couple of weeks, depending on literally what hospital system, where the patient was. And that was just really agonizing for them and agonizing for the doctors, the hospitals. We wanted to treat the patient. So there's a very good financial case as well. It's not just an emotional case. The idea of that patient getting the biopsy, knowing what they have, and being able to put them on the books for treatment immediately meant better care, better outcomes. We know that in 4 weeks, if there's a delay in diagnosis, in 4 weeks, those cells, the NIH study, can metastasize and lead to a decreased survival or mortality. So that's what we're trying to prevent. We always talk about early detection, early detection, early detection. So 10 days of waiting is not early detection.
Definitely.
And that's now that FLIM is able to do that immediate differentiation. So, however, there were a lot of engineering problems with this device. It was way too big, way too costly. You're very sensitive to vibration, things like that. It just wasn't acceptable or work in the clinical setting. And so it's never made it out of the academic research lab until now. So that's what my company did. We innovated off of this technology. We made a desktop-sized solution, and we've tested it. We're a little early on 32 specimens, but we did prove the difference between cancers, a couple different types of cancers, and normal healthy tissue cells. And we did that in a mammalian model, a mouse model, but using human cells, so human tissue. And with that, We're developing currently a basic AI database based on the parameters that we have, those numbers, lifetime numbers, other mathematical numbers, other functional numbers, and that goes into the algorithm. And this is exciting because pathology is a field that's always been structural, where the doctor or whoever it is looking at a slide and seeing something looks funny.
Yeah.
And there's all sorts of criteria for what makes something malignant and what doesn't. But what this technology does, what the RapidX machine that we devised does, is it measures that function of metabolic activity. So it is quantitative, it's objective, it's not a subjective look. We also have imaging, it does image as well. So we can add that to the database. But that's what's exciting. And that's why it's capable of doing it now, because in the past you needed a human, and not just any human, a highly trained
A
biomedical
superhuman.
engineer, right, to catalog and interpret these results. And now with the AI database, that won't be necessary. So that's what we're working on. And it really is possible. Not only is it possible, it's necessary. It needs to be done. Needs to be done yesterday.
Thank you, Rachel, for such a detailed walkthrough, and I feel like I understood everything now. So again, this sounds amazing, and it is actually groundbreaking. So you mentioned the film technology earlier wasn't as mature as to be used into diagnostic at such a level, at the level that you are able to do now. And so is it because now there is like you have help of AI and you have like help of better sensors, or so basically what was the actual challenge of implementing film into solving into a diagnostic tool basically?
Well, identifying, I would say it's like a cable box size FLIM unit was incredibly difficult. We literally searched the world and we found it. We even have the abilities to now develop that ourselves. We understand the semiconductors and how all of that gets put together. So that was a challenge that I can tell you very few people could have overcome, and we did. And I'm not, giving ourselves props. I'm just saying that, combination of very hard work and determination and some luck allowed us to find something that was small and didn't have all of those issues. Like I said, like the large FLIM needs anti-vibratory tables. When I first, started, investigating this, I was in the hospital and the research center. If I put down a pen down on the anti-vibratory table, the research fellow would be, very shocked. They take that off right now. Like there's no vibration allowed. And think about a busy clinic where biopsies are being performed. There's a lot of vibration. There's a lot of
Yeah.
people walking by and things like that. So we needed to do that and to develop a device that can be handled by anybody. So any technician, even a student, can take the tissue, load it into the machine, let the machine do its business, the RapidX. A couple minutes later, you can take it right out and put it into formalin and still have the pathologist do the check. So for a while, we have the training wheels on. We're assisting the pathologists. We're getting a fast answer for the patients. We're retaining those patients and scheduling them for surgery or chemotherapy, whatever they need. Those are high ticket items for the hospital, as well as, like I said, better treatment for the patient. The faster you can get them started, the better for their outcomes. So, that's really the goal. And again, removing that anxiety, making it so that patients can say, at least it's out, at least the
Hmm.
cancer is gone, at least we've started treatment and I'm battling. Don't make them wait a month to battle, because it does take that long. I mean, the pathology is a 10-day wait, but then after that, the scheduling and the coordination, everything else starts to become a very big deal and takes a long time. So this would change that management and make it better for everyone, including the hospitals and even third-party payers. Insurance companies hate having to, a code for this and a code for that. A lot of this starts to get bundled, So, I mean, Rapid Pathology has an existing CPT code, so we don't look at that as a challenge. You mentioned other challenges. So yes, we are starting to build a rudimentary AI database. It will be more refined when we optimize the camera for higher resolution. Our first tests were just done to make sure that the camera could get us those results that we need. But we do have a number of, like I said, numerical parameters that we can use to build the first AI database. 32 specimens. It's hard to believe. 250,000 pixels. That is 420 million AI database points. So you can see how valuable that database will be once we have hundreds of specimens, both from clinical studies and basic science models. That together, it'll be in the trillions. So if somebody down the line, even us, creates like a handheld model, for instance, that database will be critical to for any interpretation.
Rachel, so seeing how groundbreaking this is, and again, I think my reaction is, of course, when I was reading about it, that how is it possible that something that used to take like 30 days, 14 days, and now is being done in 5 minutes with the help of just updated sensors and AI. So I have like, my curiosity gave me like basic two question, and the first one, I think you've partially answered it already. What I wanted to ask is, what's the reaction of basically clinics or even the patients when they hear that, okay, a test that used to take 14 days is now being done in 5 minutes? Is it even accurate? Do you get like that kind of questions?
Well, so we do. And first of all, we have about, I would say, 12, surveys of patients saying, please put this in. We need this yesterday. Another 15 memoranda from key opinion leaders also supporting the use of this device. People see, the obvious value of it. The accuracy of the FLIM over, since like 1930 is 94 to 99% accurate, distinguishing cancer cells from non-cancer cells. And we received the same accuracy rates on our initial study. I'm not going to tell you that 32 specimens is adequate. It absolutely is not. We need hundreds of specimens. But in the meantime, while we're proving accuracy, that accuracy is going to get improved with every specimen, every minute, because that goes into that AI database. And after we initially program it, then there's deep learning that occurs. So deep machine learning, the machine teaches itself. So it starts to get more and more accurate and not only tell you cancer versus no cancer, but what type of cancer this is, what's the grade, how malignant it is, how aggressive it is, which right now, currently, what we do to find that out is we send these specimens off for expensive tests like genomic testing to tell patients whether they need chemo or not, for instance, or certain drugs, etc. So this might be able to answer those questions without the send out. But if the send out needs to be done, it can be done in concert with what we're doing. So like I said, with time, we'll see the accuracy. And, we're highly ethical as a team. If the efficacy doesn't pan out for some reason, well, accuracy rather, we would report exactly what results we get. So, and if we see that we can help in some way, but not as big as we want to, that would be something that we would absolutely report. But that's just not something that we see. Again, with a almost 100-year history of this technology working, there's no reason whatsoever why it won't work with our modification, because it's the same technology at its core.
Understood. And I think it makes perfect sense. I guess my question is also about, and actually, you're totally right. If it has worked so far, it should work now as well. And it is working so far that you have tested. So my next question is like coming from here itself. There's clear proof of that this is working, and you have data on accuracy as well. So what's the current challenge do you see convincing hospitals and clinics in adopting basically RapidX? So are you facing any challenge in that?
Sure. Well, we're a little bit early for that. I would say, we need funding so that we can build a couple of prototypes leading to a minimum viable product, which means adding some software, adding a couple of modifications on the device. The device, in my opinion, needs to be perfect. So that has to happen. And then we have to be able to, we have begun collaborating with MD Anderson in Houston, Texas. So we want to start to basically have a second device that we give out to MD Anderson and begin a clinical study. So those are expensive. The clinical studies are very expensive if we want to get high enough powered studies. So that means enough specimens for it to be significant. And with the clinical study, we're going to see a lot of heterogeneity. We expect it. We want it. We want to use all that data and program it up. But in order to have an adequate specimens of repeat things, we need a lot of specimens. And again, that is an expensive challenge. And also that has to be consistent with what the FDA is looking for. We will be socializing soon with the FDA, and their costs are also relatively high. So these are some of the barriers that we are looking at. We don't see the FDA as being an impossible challenge. We're removing tissue from the patient. There's no injury whatsoever from the RapidX device to the tissue, and there's absolutely no risk to the human. We're not doing anything different than what's already done. There's always risk from a biopsy, but we're not adding biopsies. We're doing the same procedure, maybe even less. Let's just say we take out 3 specimens and we find what we're looking for in 3, and they were planning to do 12. They can stop. So you actually may decrease the risk of bleeding or hematoma, or in lung, things like collapsing the lung. This may avert massive complications, which are costly and really hurt the hospital and the patients. There's all sorts of benefits that can be realized with this device. But
And
those are some of the challenges.
I totally agree, and I think it's better because in all all you're taking is just 5 minutes away from the process that otherwise would have been like 7 days. So anyway, if is not convincing enough the result, they can always go with the usual procedure they were anyway going to go ahead with.
Well, we will. We're not taking that away, anytime soon. We still will send that up to pathology for
Hmm.
absolute confirmation. But again, if we keep seeing it's exactly the same, we know that it will be great to tell patients that. In the beginning, the stipulation to the patients is our RapidX is we're waiting for final pathology. In the meantime, we're going to get you scheduled and do everything, and then the
Hmm.
pathology will come in. And again, we have absolutely no reason to believe that it will be any different.
Yes,
So
yes. And also thank you for mentioning, the regulatory and FDA pathways that you are planning for. So where are we in terms of, these FDA approvals or regulatory or clinical trials?
we need, like I said, I told you the couple hurdles that we're at right now. We need some clinical studies, and that data will be used for the FDA. But we expect a 510K pathway. There's a massive movement right now on pathology to go digital. So basically, the usual pathology, the one that takes the 10 days, for instance, they're digitalizing those images. AI can actually enhance the reads on those images. So the human works with the AI.
Mhm.
Basically, it's showing that it's more accurate when you add the AI to the human read. So it's helping. So that's sort of a predicate for what we're doing. And so we see a 510K pathway ahead, and maybe a breakthrough designation, because if we have information on patients and we withhold that, that's not ethical. Basically divulging or yielding and discussing that information with the patient, we think is going to be completely necessary. And we think the FDA will agree. So that's what we're expecting. At worst, a de novo pathway. We do not think we'll be undergoing a PMA, which is the very lengthy pathway for a class 2 device that doesn't inject anything into the patient or expose the patient to any harm or anything like that, or the specimen.
I think I get it, and I wish you good luck for the approval process. I see. I mean, I totally agree that this is not a very invasive process, or it's not something that that causes a lot of harm, and then FDA has to like go into a lengthy process for approvals. So again, good luck with whatever's coming, whatever the next steps are for these regulatory pathways.
Yeah, thank you. And listen, I'm not trying to minimize. It will be, a process with the
Yeah.
FDA, but we're ready for it. And the CPT, or the CMS process for reimbursement. I think we have it under good authority that because the rapid pathology, which is done today, it's not the greatest, but it's done. So the CPT code already exists. So we think we'll capture that without much of a problem, or some sort of modifier on that. So those are the biggest hurdles that startups really talk about. But we're in that zone. We're not quite in the Goldilocks zone, where a lot of venture firms or angel firms even they want to see a little more proof. So we're trying to just raise the dollars to get to that place. We know, I've seen it with my own eyes. I was there doing the first study. And it's pretty amazing. I mean, aside from all the numbers that I told you about, as soon as, I just dissected a cancer out of a mouse and put it under our camera, and it lit up like a ring of fire. It was pretty amazing. And I said, this works. This is exciting. For my team, I had my COO with me. It was our lunar landing moment. It was Neil Armstrong on the moon. So, we were just totally floored.
I
And
must be jumping, right?
yeah, being able to see that was extremely exciting for us. And we wish that the outside funders and whatever could have been in the lab with us at that moment, because it was just that exciting.
So Rachel, I think I have a very good clarity about what's happening, what challenges you have faced, and where RapidX is and where Caelum is in terms of basically growth, I would say. So apart from funding challenges, what's the scientific or technical challenge do you see coming up in future growth? Yeah.
Complete transparency, the device that we're using is very good at inspecting the XY axis. So, basically the width of the specimen and the length of the specimen.
Yeah,
But it's not great at the depth of the specimen. So, our next study is to add multimodality using the same technology actually to get cut on like a CAT scan, to get serial cuts of the specimen, or add ultrasound or some other multimodal technology that will be able to see through the entire specimen. When people say, oh, we have a problem, it doesn't this, I say, no, that's an opportunity for a patent. That's what I always say. So we did actually file a super patent with all of our little bells and whistles and, these to adopt to this and that. So that's good for the year. That contains about 20 patents within it. So we'll decide at the end of the year which patents we'll actually use.
So are patents the primary way you are protecting your innovation, or is there any trade secrets or?
We have trade secrets. We have trademarks. So yep, we do all of it.
That's good to hear. And I think with that, Rachel, I am end of the line of my question. Actually, I understood a lot of things. Thanks for taking me through the journeys in such a depth. I really like your way of storytelling. But just before we close, I have one more question. And
Absolutely.
this is something, I ask every founder, and this just like coming from my personal curiosity. So as a healthcare entrepreneur, what is going to be your advice? I have talked to a lot of healthcare startups and biotech startups, and I believe this is like one of the most toughest field to survive in and to grow in, right? And so far you are doing very good in this. And after talking to you, I can see you are like very optimistic person, and as you mentioned, you look at problems as opportunities. So what's your advice, for example, for other startup or entrepreneurs who are in the same shoes as you?
Sure. I will say, full transparency. I've been smiling and I'm optimistic, and I tend to be, but there have been some very difficult moments. I have a cousin who said, you're going to find that every couple of days or weeks, you're going to have an existential crisis. And he was completely correct. That's how it feels. There have been a lot of setbacks, a lot of challenges. So my advice is just, if you're going to do this, you've got to have that stick-to-itiveness. You've got to just prepare that the whole thing is a war, and it's battle by battle by battle. Treat yourself well. Don't self-terrorize at all. You have got something and you know it.
Yeah, you have to hold on to it.
Hold on to that belief, as long as you know it, and as long as you know it to be true. If it is not proven true, that's different. But as long as you know it to be true, you got to hold on. And if you want to do something that is highly disruptive, you got to put your seatbelt on. It's going to be a ride. So just that's full transparency. Things that are less disruptive or a little bit easier to do, a lot of value in a lot of different things. I'm not knocking anything. I'm just saying that, something that is, like I said, highly disruptive is also going to be a harder mountain to climb. So just be prepared. It will take longer than you think. It will cost more than you think. But if you really stick to it and you really hold on to your beliefs,
You'll
it
get there
you
eventually.
will get
Thank you, Rachel, for, and I think your advice, I believe it applies to everyone. It applies to me as well, not just any healthcare startup. This is a very good advice. So thanks
that's
for,
good advice for life. If you feel that you have a purpose and that you've been called to it, you have to pursue it.
thank
So
you so much for sharing that. And with that, Rachel, I think we are end of our interview. It was amazing talking to you, and I got to know so much about it.